Welcome to the Bio21 Institute and to this Convergence Science Network event. The Convergence Science Network acknowledges the traditional owners and custodians of the lands on which we meet today, the Wurundjeri people of the Kulin Nation. We pay our respects to the First Nations people and acknowledge elders of the past, of the present and of the future. I'm Luan Ismail, the founder and director of the Convergence Science Network, and I must say it's just wonderful to have so many people in a room for an event, and it's been a long time between drinks, as they say, so I'm very, very grateful for your support to be here this evening. We have a very interesting discussion. The Convergence Science Network has held 4 events on this topic over a period of about 4 or 5 years. And we had a huge privilege in 2018 to host now Nobel laureate Jennifer Doudna, who kicked off this series, if you like, discussing the ethics of gene editing. So I'm delighted that we're able to bring together an excellent panel today to discuss what is continues to be a challenging and hard issue. And of course, the last few years have unquestionably been and reminded us that the biomedical sciences can be controversial. There was a 3rd International Summit on Human Genome Editing held in London a month ago, and the organising committee made the following statement at the conclusion of the summit. Heritable human genome editing remains unacceptable at this time. Public discussions and policy debates continue and are important for resolving whether this technology should be used. Governance frameworks and ethical principles for the responsible use of heritable human genome editing are not in place. Necessary safety and efficacy standards have not been met. Governance mechanisms for human genome editing need to protect ongoing, legitimate research while preventing clinics or individuals from offering unproven interventions in the guise of therapies or ways to avoid disease. I'd now like to ask you to watch the screen as we play a short video compilation to provide some context for the discussion that will follow.
CRISPR-Cas9, bring me a gene, encoding for a specific protein. Make a few snips at this coded locus. You work so well inside a streptococcus. Cas9, I'm so alone, without your scissors in my chromosome. Cut me up and do it clear. CRISPR-Cas9, bring me a gene.
The doctor who sparked global outrage by claiming to produce the world's first genetically modified babies faced his peers for the first time at this conference in Hong Kong.
As you can see, there's a big demand to know exactly what you did.
How you understand your responsibility to these children.
He was grilled by bioethicists, regulatory experts, and fellow doctors, but he remains sketchy on the details of his experiment.
I don't know how to answer this question from my eyes.
There has been a failure of self-regulation by the scientific community.
Shenzhen-based He Jinkui used a gene editing technology called CRISPR to engineer twin girls whose DNA was designed to resist the HIV infection.
The world has moved on to the stage for embryo genome editing. There will be someone somewhere who is doing this. If it's not me, it's someone else.
The news of the birth startled the scientific community.
The general feeling amongst the scientific community is that we shouldn't go ahead yet, but you went ahead without really discussing with people. That's how it seems.
I also consulted with several experts,
as someone who's been involved in the genesis of the CRISPR-Cas9 gene editing technology, I feel deeply disturbed. It's inappropriate and something that we have been working hard to avoid.
Why you choose to cross this line?
What is our responsibility as scientists and doctors?
May we know your future medical care plan for these babies?
How did you convince the parents?
Scientists here are worried about the safety of the CRISPR technology, and many cast doubt on whether Herd really did what he claims. His experiment would be banned in many places such as the US. For his part, Herd says he supports using gene editing for diseases and not for improving humans. He also claims that there is a second woman carrying a genetically modified baby in China.
Are there any current pregnancies with embryos that have been genome edited as part of your clinical trials?
There's another potential pregnancy.
You may wonder why we use the gene surgery to prevent HIV. Why not another lethal disease? Two reasons. First, safety. Second, real-world medical value. As a scientist and the father of two girls, safety is my #1 concern above all else. The gene we choose, CCR5, is one of the best studied genes. In fact, about 100 million people naturally has genetic variations that disable CCR5, protecting them against HIV. They were healthy. Our surgery caught exactly at the start of that natural variation. Decades of clinical trials has tested medicine to mimic that natural variation, including the first gene editing trial in the United States. Well, it proved safe. Also, protecting against HIV requires the simplest gene surgery, which is to remove just a few DNA letters. Correcting a broken gene will helpful for more diseases like familial cancer or muscular dystrophy. It's more complex. Studies in the US hasn't shown this works well in embryos yet. The second reason we end up choosing HIV as our first target is the real-world medical value. HIV remains a deadly and devastating infection that needs medical advances. About 1 million people died of HIV last year. New infections remain 3 times higher than the UN AIDS target for 2020. No vaccine or cure exists. Taking pills daily from life carry healthy risks. In many countries, discrimination deepens the devastation of infection. Employer may fire people after discovering their HIV status. Doctor may refuse to treat. CCR5 surgery, while not applicable to the general public, it may be valuable to help a few affected and very high-risk families protect their children from the same fate. This led to the National Academics in 2017 named vaccine-like disease prevention as an ethical application of gene editing.
All right, so I'll end by simply noting that while much work lies ahead to fully realize the promise of therapeutic gene editing, I'm optimistic that the coming years will bring much of this promise to patients. I suppose that's the reason I was invited to be the hopes part of this hopes and fears session. I'm hopeful because, quite simply, the relentless use of our collective efforts, gifts, and resources to improve our condition and those of our children is perhaps the defining trait of our species. While therapeutic gene editing technologies have become widely appreciated over the past decade, they are actually the fruits of an ongoing 70-plus-year-old quest to develop one of the most fundamental capabilities of humanity, the ability to have some say in the sequence of our genomes so that we are no longer so beholden to the misspellings in our DNA. Thank you for your attention.
So without further ado, I would like to invite Professor Megan Munsey to come to the podium and host the discussion. Professor Munsey is a developmental biologist who leads research programs into ethical, legal and social implications of stem cell research at the Murdoch Children's Research Institute, the University of Melbourne and Renew, the Novo Nordisk Foundation Centre for Stem Cell Medicine. Thank you.
Thanks so much, Luan. And I'd like to thank Luan for getting us all here tonight. And I think it is wonderful to be speaking in front of a live audience. I'm sick of Zoom, I'm sure you are as well. Sorry for those who are watching the recording, but it's really nice here. And I think it's a really important topic, as Luan mentioned, and as is championed by those who've been gathering in London at the 3rd International Summit. We need to have these conversations. We need to have forums where we can have frank and fearless conversations about new technology. So I really welcome and again thank the Convergent Science Network for hosting tonight and for starting this conversation. So I just wanted to add my acknowledgement to the Wurundjeri people and their elders past and present, and of course any Australian Indigenous Australians who are joining us here tonight. I'd like to pay my respects. We've just heard at the end of this video that Luan has compiled from David Liu. He's a researcher, and as he said, he was invited to speak about the hopes of the technology. And I was really struck by that quote he used, that perhaps through this technology, no longer are we beholden to the misspelling in our DNA. I think while this relatively new technology could potentially alter how we use and identify and perhaps prevent disease in our community, I think changes to the DNA of every cell in the body of a child carries with it substantial risk and substantial challenges that I don't think are necessarily yet addressed. And we'll come back to perhaps some of the quotes from Dr. He in a moment. But what I want to do tonight is to talk about whether we could or even should use this technology and what it means in the Australian context. And I'm really delighted to be joined by 3 fantastic panellists. I'm going to introduce them each in turn, and they're going to come up here and take a seat. And we're going to have start a bit of a conversation around some of these, the ideas that have been sparked perhaps by the video and by some other questions that I'd like to ask them. And then we're going to open the floor to you. So start thinking about your questions, and then when I turn to you, just put your hand up, introduce yourself, and ask our panel what you want to know, because I think that's the important opportunity here. So I'd like to introduce our first panellist. Come on down, Professor Diane Nicol. Di is
like I'm on a game show.
I know, that's what I'm trying, that's the vibe I'm going for. Di is Distinguished Professor Emerita? My Latin is appalling. Emerita. At the University of Tasmania, and she was formerly the Director of the Centre for Law and Genetics. Our second panellist, come on down, Professor Rob Sparrow. Chris, sorry to give you eye contact there and then just disappoint you. Rob is Professor of Philosophy in the Philosophy program at Monash University, and his research interests are political philosophy and applied bioethics. Our third panellist is Dr. Chris Gyngell. Chris is a Senior Research Fellow in Biomedical Ethics at the Murdoch Children's Research Institute, and he's also the Co-Director of the Health, Law and Ethics Network at the University of Melbourne. So I'm going to come and join you over here, and I'm going to ask the first question really to Di. And I'm going to start by saying that in that montage we just saw, you and I were sitting in the audience together.
In row 3, I think.
In row 3, right up the front. Right up the front we were. I just wondered whether you'd like to start this evening's discussion with reflecting on what your reaction was when you heard the news about Lulu and Nana.
My first reaction was shock and horror. How could somebody at this stage of the technology even contemplate doing these things? Irrespective of whether we should in the long term or not, the fact that this person had done this at that moment in time just seemed wrong to me, to create children without any knowledge of what the consequences would be. And a lot of the concerns, I think, have panned out now. Embryos are really, really complex things. What this research has shown is that weird things happen. It's not just simply knocking out a couple of bits of DNA. There have been large-scale deletions. There have been all sorts of other things going on. So it just shows that we're just so not ready to do this on human embryos that we're going to implant into women and have live babies. If it's ever going to happen, it's certainly not going to happen right now.
And I think that issue of safety, we just saw from the interview or the pre-recording of Dr. Her to providing a justification, and safety was paramount. And yet,
there we were.
You would challenge that?
I would, and it was quite interesting sitting next to some of these people. I after the session, I actually took a taxi to the airport with Feng Zhang, who is one of the leading proponents of this technology. And he said some of the graphs that were shown really showed that this was not working, that there was, one of the risks is called having chimeras. This means that some of the cells are changed and other cells aren't. And he said he could see from those slides that there were things going on that just didn't seem to make sense. So safety is a real concern, apart from all of the other concerns that we will doubtless get into.
Well, maybe you could pass the mic to Rob, and if I could ask Rob to perhaps reflect on his thoughts when he heard the news.
Look, in some ways, I sort of expected this. This technology is always going to involve heroic medicine. There's a real drive to be first here. I'm in a strange situation reflecting on this technology because I'm deeply critical of heritable germline genome editing. But I think the arguments about safety are actually a nonsense. I'm just going to go out there and say, you're never going to know whether this technology is safe. It's always going to be experimental. The first time someone does this, you could say exactly the same things. It won't matter how much animal research you've done. The first time you do it in human trials, we won't know whether it's safe or not. And so it seems to me what people are criticising Dr. Her for, we'll be able to say that in the future. We'll be able to say, whenever this technology is done, we'll be able to say, how can you possibly know whether this is safe? You're experimenting on people. So that's one reason for thinking that the safety arguments, I kind of think they're a red herring in this context. I think there are real reasons for concern about this technology, and I think you can say it's unsafe, but I think there's always going to be real doubts about its safety. I also think you need to put this technology next to, say, the decision of parents with a history of familial genetic disease to have children. Because there's a sense in which that's unsafe. There are some people, reproduction is unsafe in general. Terrible things happen, it's risky for parents, terrible things happen to your children. You're always risking the health of the future child when you reproduce. And some people, given what they know about their own genetics, are actually taking a very big risk with the health of their future child. So it seems to me if you're going to criticise this technology, you might have to start saying there's people in the community who shouldn't be thinking about having children because it's too risky for those children. Alternatively, you can defend those parents' reproductive liberty and say we're all taking risks when we have kids, in which case this doesn't look so unprecedented anyway. And then there's some further philosophical complexities about whether or not those children would have existed except for this technique, and whether you can compare their circumstance with what would have happened what their welfare would have been like had these experiments not been performed, they certainly wouldn't have been any better off. So the comparison with non-existence makes making assessments about the ethics of this technology really difficult. And then I think you just want to look back on the history of IVF, ICSI, all reproductive technologies, heroic experimentation on future children. There's a sense in which we still don't know whether IVF is safe or not. At the age of 70, every child born of IVF might spontaneously combust. I don't think that's very likely, but we don't know that that won't happen. So if you don't think we should be taking risks for the health of future children, that places reproductive technology in a real bind. So, I mean, there's obviously more that can be said here, but I wasn't surprised, I guess. And I think that the arguments that we are rehearsing now about risk and safety are precisely, people will just have this debate again in 5 years or 10 years, and they'll have a few more animal models and they'll have a better understanding of the genetics, but they won't know what the impact of this technology is on the future children.
We'll come back to that safety issue and what possibly could be done and explored. And I think we might also come back during the question and answer session around IVF and safety, because I know there are some people in the audience who have strong views on that. So perhaps we might, and some experience, we might come back to that. But Chris, I want to get you to now grab the microphone and maybe just reflect on how the news of Lulu and Nana struck you. And I know this is an area that was not, again, new to you. You'd been thinking about it for some time. So what do you think some of the ethical issues were?
When I first saw it, I thought it was a hoax, because I first saw He Zhonghui on YouTube announcing the future of medicine, and it had this production feel, like this cheap production, and I thought it was a hoax. I didn't think someone had done it, but obviously they had. And, I think it was very, very surprising that at this stage it had happened. And when you watch the film of him, I think he thought he was going to win a Nobel Prize. He talked about the inventor of IVF and how that went to Nobel Prizes. So as Rob was saying, with the heroic medicine, it seemed like that's what he was doing. He saw himself as a pioneer. I was surprised that it happened, so quickly. But, I've probably got a different background to Rob, that I think a lot of problems of this technology involve timescales, and I agree we should go slowly, but I think if you broaden timescales to 50, 100, 150 years, this could be a really important technology to ensure human flourishing. We know that a lot of people die prematurely for no good reason, and it's unfair, and as an ethicist, I think it would be better if we all lived healthy, long lives until we're 80 or 90. So I think we should be developing this technology to achieve that goal one day.
So keep the microphone, and I think you're absolutely right that Dr. He did talk about wanting to be the first. That was certainly something he said on the stage at the conference. But this issue of how do we advance the technology? So you've said we should. Yeah. So what are the next steps if we were to?
I think when you're doing research ethics, it often comes down to risk versus benefit. I think one of the reasons the He Zhonghui experiment was so atrocious is because you've got embryos that, although HIV is a serious disease, it's not a deadly disease in infancy. And it's a treatable disease. We don't know what HIV treatments will be like in 20 or 30 years. You might not, might not matter if you're susceptible. You might be able to treat it equally. But there are catastrophic genetic diseases that you could do experiments on to see the safety of the technology. For instance, there are genes that make it impossible for a fetus to develop, essentially. So single gene mutations, and if you have that, you'll have a miscarriage, you won't have a healthy baby. I could see how you would start by looking at those catastrophic conditions and trying to see what happens when you do gene editing to see if that does correct the issue. After you get the embryo ones, we know that there are lethal, untreated diseases in childhood, like Tay-Sachs disease, which are really dreadful diseases, which you see babies die in the first months to years of life. Even if you did gene editing on that and say it was wrong and there were unintended consequences, you're not essentially harming that child much because they were going to die very, very, very soon. So I would see it, if you're going to start to develop this technology, to start with those cases where the risk-benefit ratio is much, much clearer.
In that case you just mentioned, that would be performing genome editing on a child. So quite different to...
Well, you could do it at the... So I'm thinking of a case where you have family history of disease, they go and do IVF, they have one embryo, it's got this disease.
Right, so it's identified.
It's identified, yeah.
Yes, Rob?
Sorry,
so we have to share the microphone. Sorry, it's a little bit cumbersome.
So I do agree with you that the choice of HIV just seems bizarre in some ways, and the idea that there are families, who are particularly high risk of HIV, that also seems to me quite a strange claim. I guess the obvious thing to point out here is people who want to become parents of healthy children, insofar as anyone can guarantee that their child will grow up healthy, can always use donor gametes. So when we're thinking about the therapeutic potential of this technology, you're presuming that you have a prior strong moral reason to want your child to be genetically related to you. Because if you just wanted to have a healthy baby, you can use donor gametes, and you or your partner can give birth to a healthy child, or as much, again, because there's no guarantee of anything in this world, but a child who doesn't have the sort of lethal disorders that you're concerned about. So yes, you could do this technology, but what you're really serving is the parent's desire to have a genetically related child. This is not about having healthy babies. There's a prior argumentative move which says, I'm prepared to experiment on my child, radically experiment on my child, because I don't want to give birth to a child who isn't genetically related to me. So yep, it's only therapeutic if you think there's no other way you can have a healthy child, and given that we can use donor gametes, you can have a healthy child. So this is a very boutique technology, and it's reinforcing a message that genetic relatedness is so important that we will do this really radical and unprecedented thing, and that giving birth to a child that was conceived using sperm or egg from someone who's not related to me, that would be worse. So it's only therapeutic. It's really curing a parental anxiety about a lack of a genetic relationship with the child. So I just can't see a strong therapeutic case here.
And I'm going to get Di to take the microphone, but as we're transitioning, I just want to say that I'm glad you corrected that assumption also, that use of donor gametes would necessarily guarantee that the child will be without genetic disease, because it may not. No. Di?
And of course there is the technique of pre-implantation genetic testing as well. And so the people who might actually really need gene editing to have a genetically related child for serious disease, it's really a very, very small number of people. Yet one of the concerns I have is that so much of the debate about gene editing is taken up by this debate around heritable gene editing. And to me, there are so many other aspects of gene editing that need to be discussed as well. So, for example, there are a whole array of clinical trials that are currently going on for the treatment of children and adults who have genetic disease. And I think that it's interesting to see how they are progressing, but they themselves raise a number of ethical, legal, and social issues, which we're just not discussing enough because we're so consumed by this issue of heritable human genome editing, and that's somewhat frustrating, I think.
So, given you have the mic, I wouldn't mind you just elaborating a little bit about that, because I think that distinction is really worth making. And I know at the second summit that we were at, that was what we were there to talk about. We were there to predominantly talk about what we call somatic cell genome manipulation, whether that be by delivering a gene vector directly into the patient, so in vivo, or taking up their hematopoietic stem cells, their blood stem cells, and manipulating those in the lab. So what do you think that the kind of, you mentioned that there are regulatory and sort of ethical, social issues associated with this somatic cell manipulation. What would you say they are?
So this is the area, as I say, where I think there are really interesting developments going on. And as Megan has said, there's a big distinction, I think, between heritable genome editing that actually affects your germ cells and can be passed on to future generations, and these somatic cell changes, which are only within the child or within the adult, unless there's a mistake and somehow they get into your germline cells as well. And one of the really interesting developments is for a disease called sickle cell disease. So sickle cell disease is a blood-borne disease. It affects the shape of the red blood cells so that the red blood cells can't get enough oxygen. And it primarily affects people of African descent. This is almost at the point of being approved as a therapy in the US. And to me, this is such an important step because it's actually benefiting members of society who are usually the more disadvantaged members of society. So many new drugs and so on are for Western diseases. And to see something that is primarily focused on people from African countries, I think it's important, and it does raise important discussions around equity and distributive justice and so on. One of the things that's discussed a lot is that the research that's going on in gene editing, but in genetics generally, it's primarily focused on people of European descent. So we simply don't have the research that's going on with people from other areas of the world. And so inevitably that leads to disadvantage. And those are really important discussions to be had. How do we actually achieve justice on a global level? How do we make sure if gene editing for sickle cell disease is available, how do we actually deliver it to people in Africa? It's going to be really, really challenging, but it's important, I think, to have those discussions now.
Oh, thank you so much for elaborating on that. And I want to go back to inherited change in a minute, but just before we do, just whether Chris or Rob would like to add anything to Di's comments on somatic cell manipulation.
So, I mean, those points are well made. One advantage of the somatic cell modifications is you can often ask people whether they want to undergo the therapy, so you're not necessarily experimenting on children, though of course you could do it on infants as well. It is worth mentioning the limits of that technology, which is it's limited in the number of cells in the body that can reach often, and so the kind of embryonic manipulations that will tend to be heritable are more powerful in terms of being able to change perhaps every cell in an individual's body, and that's one reason why scientists are keen on this stuff. I mean, I do think it's really important to understand that this is a tremendously powerful and exciting technology with all sorts of applications across, fundamental science and medicine that we should expect wonderful things from. So I think that focusing on the heritable germline genome editing, it is important to say, okay, maybe, as I would argue, that's a step too far, but there's all sorts of other research using these fundamental molecular techniques that I think are, we've got to hang on to and look forward to. I mean, the other thing to mention here is what's happening to our relationship with plants and animals, because the commercial application of this technology is also going to be in animal husbandry and xenotransplantation, and so there's applications in non-human species that it's worth keeping in the back of your mind as well. And some of those again are probably, growing replacement organs for people is going to be quite in genetically modified animals that will turn out to be quite hard to argue with. It seems to me. I mean, again, you can make some arguments about that, but there's going to be a very compelling case for organ creating replacement organs using something like this technology. So there's a lot going on in this space, and it would be a terrible mistake to respond to the applications in human beings with a wholesale rejection of the use of this set of molecular tools.
Yeah, just to tie it back to the heritable gene editing, because, I think that's right, the sickle cell development is a really wonderful development. I think it's a bit optimistic to think it's going to have a huge immediate impact in Africa because of the cost. It costs around $2 million for a treatment for one person. Just to get it back to the heritable thing, just think about what will happen. So you might, we've got this sickle, it's a brilliant invention. We take people's blood cells out, we gene edit as many blood cells as we can, thousands, millions of blood cells at a tremendous cost, but we don't change their germ cells. So that person can have a happy life. They'll have a family. Some of their family will probably inherit sickle cell. They'll get the $2 million treatment over and over again. You could just edit the cell when it's an embryo stage. One cell at a tiny fraction of the cost, and that will see that none of those future generations have to have this incredibly expensive gene therapy. So, I think somatic therapies are good, but I think we have to think about all the tools in our arsenal to try and create a healthy future.
Could you use PGD to select for an embryo? Do you have to still do editing for sickle cell anemia?
I guess so. I'm not a scientist, so but I guess that's right. We maybe could.
So
but
again, that's another
it's another tool.
technology.
It's another tool,
And
and if
it's
that's a
also
cheap tool that's effective to reduce rates of sickle cell in the future generation, I would encourage it.
Yeah, and I think it's again a question of access, because it still is a technology that is going to be difficult to roll out in Africa. But
Yeah.
Rob?
So again, that whether you can do that depends on the genetics of that condition, which I'm not across. I do think it's worth keeping in mind, if you're serious about people's health, how much you could do with environmental manipulations that we understand how to do now. I mean, there are equity issues to do. A technology that costs $1 million or $2 million to cure a single individual. You think, well, how are we going to roll that out to everyone? People need clean drinking water. They need public sanitation. They need not to be displaced by civil war and climate change. If you were serious about thinking about the health of people in the global south, it's not obvious to me that you'd be talking about these kinds of high-tech interventions at all. And the sorts of concerns that people have about distributional justice and over-investing in these technologies are also kind of too powerful, in the sense that you can say that about heart transplants, or you can say any sort of, almost all of the medicine that we have access to in Australia, for which I'm deeply grateful, is medical care that is not available to people in large parts of the world. And so if you can push this argument both ways, because you can say, nothing for anyone except for everyone, and then we have to give up an awful lot until the rest of the world catches up. Or you might accept that these things are going to be rolled out gradually and in ways that seem unjust, in which case people in the wealthy northern countries will get the experimental gene therapies, and it'll be a long time till anyone else gets them. But in a sense, what you should be thinking about is, if you cared about, people having healthy kids in the global south, there is a lot that we can do that doesn't involve any genetic manipulation at all. And again, it's not obvious that stopping this research means those public health initiatives take place. But when people start talking about an urgent moral imperative to kind of heal the sick through this radically experimental genetic intervention, you might say, look, I take that urgent moral imperative seriously, and that's why I think we should stop extracting wealth from the global south and immiserating people. That's why we should do something about climate change, and that's why we should pay attention to issues of food justice. Once we've got that stuff worked out, then we might start worrying about who gets access to germline gene editing.
But tonight we're here to talk about human heritable genome editing, and I want to go back now to talk about the Australian situation and the law die around, could I do this work here? What happened to Dr. He in China, and would I have a similar fate if I was to dabble in this technology?
Mm-hmm. So this is such an interesting question. I was actually invited to that summit to talk about the law in Australia. So we had a session where there were people from various regions of the world talking about what is and isn't permissible. And it was really interesting to sit there and hear this. And the simple answer is absolutely not. This could not have been done in Australia. So we we have laws in Australia that really came about when people started getting concerned about human reproductive cloning. Remember Dolly the sheep? Dolly the sheep was the first cloned animal, and this set out alarm bells, I think, globally about what we're going to do, what sort of governance we want to make sure that reproductive cloning doesn't happen in humans. Again, it probably would have been a long way off anyway, but there was a real feeling that we needed to have laws in place to make it absolutely clear this shouldn't happen. So we have legislation called the Prohibition of Human Cloning for Reproduction Act, which prohibits human cloning, but it also prohibits a whole array of other things as well, including making heritable changes to the human genome. And the maximum penalty for this is 15 years imprisonment. Not surprisingly, there haven't been any prosecutions in Australia on this, but it really shows that from the Australian perspective, certainly for the time being, this is not going to happen. Because who would do this sort of thing in Australia with that risk of prosecution? We do have other laws in Australia that do allow for certain forms of research involving human embryos. But again, that's really strictly limited and strictly regulated. So in Australia, I guess, depending on one's perspective, you can sit back and say, we have this regulatory regime in place, which is really strict and really is not going to result in anything like the He-Jiankui scenario.
So I just wanted to make a quick comment on the law while it was on topic. So one of the sort of striking things, well, not striking, so I think one of the really more interesting questions ones we talk about when in the future, when it's safe, what should we be able to do? Where are the limits of the technology? What should we be able to use it to do? But the really practical questions right now, the most important questions right now are research. We're trying to do research for this technology. It's not just for future heritable uses. You can learn a lot about the embryo and the embryo development by doing these things. One of the striking statistics to me, I think it's about 30% of fertilized human embryos make it to the first trimester of pregnancy. In other apes, that's like 90%. And we don't really know what it's like. And we can do interesting experiments with CRISPR gene editing to help unlock this. With the statement from some of the organizing committee, it talks about basic research. It talks about the importance of basic research, and it says this work should continue. So we seem to have consensus that research should continue, but in Australia that is illegal. It's illegal. Well, it's very, very murky. It's very not clear. It's basically the language of the law. It's illegal to intentionally make heritable genetic modifications, which a lot of people will interpret as banning this type of research. So I think there is a good cause that our laws should be reformed to at least match the international consensus that basic work in embryos is important.
So I'm going to hand back to Di in a sec, but just while you've got it, Chris, are there other jurisdictions where this has been happening? Is Australia behind?
I think the UK has a really good legislative framework which allows this research for 14 days, just purely for research purposes, not for
And again,
it's
sort of like a hard delineation between research and research to understand perhaps how the embryo forms.
Yeah, it's just basically you can't implant it into a woman and it's got to stop after 14 days. So do what you want for 14 days, can't implant it to a woman, end the experiment after 14 days.
So a very similar delineation around the cloning technology, so around somatic cell nuclear transfer, allowed for so-called therapeutic research investigation, but not allowed in terms of reproductive.
Exactly.
So Di, I could see you are dying to say something.
I'm sure you're going to start asking me questions on this soon, Megan, but I want to come in here. So we have these laws in Australia, and the thing is that if we were ever to contemplate changing the law, as has been suggested, this to me is not a trivial exercise. And what we've seen with these laws is that there was a huge amount of community discussion before the laws were put in place. And there was a huge amount of discussion amongst experts as well. And there was a huge amount of discussion in Parliament. The law that allows certain research involving humans, the debate on that was one of the longest ever parliamentary debates. It's a really huge question, and I know there'll be those of you in the audience who would think that it was totally wrong ever to contemplate doing research on an embryo because that would inevitably lead to the destruction of the embryo. So there'll be sectors of society who really strongly hold that view. There are other sectors of society who see the excitement of the technology and feel that laws should not really get in the way of technological development. Those broadly differing views are expressed across Australian society. I know this because we've done research on this and we see the diversity of views. So we need to talk to the community. We need to have these engagement exercises with the community. We're not going to get clear answers because these views are so strongly held. I don't think people are ready to change them, but it helps for parliamentarians and policymakers to understand the values that underlie these views and how we work with those values to, I guess, reach the best compromise that we possibly can. It's never going to suit everybody, but it's really important to have those community discussions, I think.
Thanks, Di. Rob, did you want to make a comment?
So, I mean, I thought that was very balanced and sensible comments from my colleague here. I guess just picking up that question about whether laws should restrict scientific research, seems an answer to that is straightforwardly yes. I mean, not just they do restrict scientific research. Think about intellectual property law here. Think about patent law. Think about how it would impact on, if you just said, look, no one can patent anything, you're not going to have any intellectual rights in your research, that would really throw the cat amongst the pigeons when it came to investment in these technologies. It's really interesting to think about when people become anarchists. Okay, and it's like people want all sorts of things to be regulated, typically what other people are doing. But all of a sudden they want to be self-regulating when it comes to the thing that they're passionate about. And think about national security legislation, all the restrictions that are going into place around research relationships with China at the moment. Research is always going to be legally governed, and that seems entirely appropriate to me because these technologies are world-changing, and so the community deserves to have a say in the matter. I mean, I guess the other thing to say here is I mean, it is striking how the moment you start talking about safety and risk, you don't talk about the other fundamental moral questions involved with this technology. There's a whole other debate to be had here about the right of one generation to impose its will on another generation, the way in which if you were gene edited, you would be marked by kind of temporality and a relationship to the designer. You can imagine in the future people will have a conversation at a party, I'm a Microsoft 2.1 baby. I'm a Microsoft 5 baby. You know, my genes are that much better than yours, because as this technology progresses, the genetic modifications that people will have access to will improve. So there's a whole lot of other stuff going on that we're not talking about here. But it does seem to me that if you're opposed to this technology, prohibiting research on it's a really good idea. Because if you're thinking about safety, what will happen is the scientists will say, oh, it's not safe, we need to do more research. And they'll do the fundamental scientific research, and then they'll perform the experiment. So if you are serious about saying don't go down this
argument.
I think the description of this argument as a slippery slope argument is itself a bit disingenuous. This seems to me straightforward to me. If people say we can't deploy this technology because it's not safe, let me do fundamental embryo research, is it going to be any surprise if in 10 years or 15 years they say, oh, now we think it's safe? And when they say that, it'll still be experimental. So it seems to me that if you are someone who is, I'm not a big fan of the no destruction of embryos stuff. That's not personally me. And a lot of people are motivated in this space by a desire to preserve the dignity of the embryo. That's not the argument I'm making. But if you don't want to live in a world where Elon Musk's children are genetically modified out the wazoo, okay, the best way to prevent that world from arising is to resist the human embryo research. So maybe not the best way, but certainly it certainly seems to me that one of the things that's going on here in this debate about whether we should be able to do fundamental research on embryos is actually a different hope for the future. People who want to prohibit that research hope not to live in a future where parents are forced to confront a decision, what gene edits do I want for my child? People who want to promote that research have in the back of their mind that they might cure genetic disease or undo the genetic lottery. Now that said, I do credit there's some pretty fundamental research, fundamental science about embryos that could be carried out using this technology. But I think that if you're serious about resisting this technology, and not everyone is going to be serious about resisting this technology, making it harder to develop it is quite a sensible policy choice.
And yet that would then place Australia well outside even the capacity to participate in the research. So we have to just accept that. And
other people are dealing smack. Should I do it? I mean, again, if you think this is fundamentally immoral, the fact that other people are doing it and will make money from doing it shouldn't actually cut much weight. Okay, that argument, someone will do it, therefore I might as well do it. There's all sorts. If your kid says, look, you know, if I'm not the one selling crack at my high school, someone else will do it. You know, there's a buck to be made. You know, it's a great argument, nice piece of philosophy. You say, look, it's immoral, don't do that.
So Chris, I can see you'd like to make a comment, and then I'm going to open up the.
I think we're just going to be so. You know, I said before, you might want to do this research, have nothing to do with gene editing. You might want to do it to improve IVF rates. I assume you're in favour of improved IVF rates, and this is the thing. There's thin. So you know, I just think it's right now the law. I think first of all, it's very, very poorly worded, because if you ask 20 people what does the law permit and what doesn't allow, you get 20 different answers because it's very poorly worded. And I think it's really important to do early experiments on embryos, which are we do allow. Right now, you're allowed to do experiments on embryos in Australia. You're just not allowed to do it with one particular technique, which is gene editing. And I just think that that's an inconsistency in the law. And you know, we should be. Yeah, I mean, that's basically my point.
Excellent. All right, now to you. Do you have any questions? Please raise your hand and a microphone will magically appear.
Hi, I'm Alan Charlson. I think it's fundamentally unreasonable to say the world scientists want to do these kind of things, because there has been one case of it, but the scientists have stood up all over the world in all these societies and agreed this was completely unreasonable and should not have been done. So, you know, I'm not so concerned about whether scientists would do these kind of things. I think there are other situations which you haven't mentioned, and the gene push technology, which is used now to control animal populations, undesirable animal populations in islands and places, is being utilised very effectively. And so there's nothing stopping people using that kind of technology to go through the whole population. What happens with gene push technology is you introduce an infertility gene in the population and it's pushed right throughout. So you eradicate rodents from islands, or you eradicate cane toads and so on. So, you know, there's some reasons to do it. But what would the situation be? What sort of situation do you think it would be desirable to edit the human genome? And maybe an example might be, you know, we've just been through COVID, and we know that there's now infectious cancers. We have that example in the Tasmanian devils, right? Now, if this hit the human populations and you could not stop it, you know, you could not stop it except if you introduced a gene, Would that be something that you would consider that you would use? You would sort of introduce a gene into the population to try and prevent that? Because we know historically human populations have been through those incredible squeezes where very few have come out. Population, again, rinderpest and smallpox are really good examples in Africa where very few species, animals and species survived and then came out because they had the genetic capacity to survive those infections. You know, what would happen under those circumstances? Would you think it'd be realistic, a government or someone to say, look, let's get this gene introduced to stop it, otherwise it's going to basically wipe us down or wipe us out? So, you know, and you could do that. Yeah, that's right. I'm just going to say you wouldn't use the sort of technologies you're talking about, but you would use gene push technologies to do that. Okay?
Look, gene drives or gene push technologies are really interesting, and the environmental use case for them is compelling. I'm not. Again, those are very modifying wild populations by driving a gene through that population. There's all sorts of ways in which that can go horribly wrong. And if you look at the history of biological controls in Australia, you should be pretty cautious before you think you're going to solve some environmental problem through genetically modifying a whole wild type of animals. I can't imagine any plausible use for a gene drive in human beings, just because the generational replacement time in human beings is way too slow. And to be frank, I can't imagine having a realistic impact on any environmental or epidemic case through this technology, again, because it's too experimental and too slow. If someone offered you to genetically modify your child now, how long would you have to wait to see whether it had worked or not? Okay, so you might think, oh, great, my infant is doing all this new stuff, you know, turned blue or, you know, glows under UV light. I mean, there's all sorts of things that you could establish in the infant, but a meaningful change, you've got to wait until they're an adult. And then you discover it probably doesn't work. So it's not a kind of, this is the germline gene editing is going to be a slow proof of concept of that. It will tend to be quite slow, it seems to me. But again, I think people will do the experiment. I have, there's so much enthusiasm amongst philosophers, and I mean, you're right, the scientific community was very quick to say, no, this is too early to do the experiment. But if, you know, George Church had done it at Harvard, would people have responded? And with a little, you know, chosen a better case, as Chris suggested, it seems to me people might have responded quite differently. So, I mean, I think that people are they'll find a charismatic couple who've tried to have children, been unable to have children because of some sort of genetic disorder. Say, we've studied in animals, you know, we've got more information about embryos because we've done the fundamental embryonic research for 5 years, and then I think some scientist somewhere is likely to perform this technology. Whether that will affect the rest of us or not, you know, maybe not so much.
Chris and Di, would you like to respond to Alan's comment?
Just very, very quickly, just back on the animals. So I think it's always important to look at the cost and benefits of different options. So right now we do try and contain animals in Australia. We try and kill cane toads, we try and kill lots of things, and we poison them. We put these poisons which have this other effect on the ecosystem. And don't just poison the animals we're trying to kill, they kill other animals as well. So one of the advantages of something like a gene drive, it is maybe better than the other options that we've got on the table, and that's my understanding of why they're trying to use it in that setting. It's a public event. I'll just quickly explain what a gene drive is for people that aren't familiar. So often when you do gene editing, you might put it's a CRISPR molecule that edits a gene. With a gene drive, you also have a CRISPR molecule there. So in the next generation, it does the same edit to the other chromosome. So basically it ensures it's inherited. So there's not the normal segregation where it's like a 50% chance. So this allows them to quickly sort of spread through the population. And, you know, I agree, it's very hard to see how there'd be a use case for that in humans for very long time.
So I don't have much to add, but I'll just say that if it was a sort of catastrophic epidemic scenario where there is the potential for gene editing or gene drives to be used, it wouldn't be to introduce it in the heritable context, it would be in the somatic context to keep people alive in the short term so that they can continue to reproduce in the future, rather than thinking about introducing something that might work in their offspring.
Or people would use the same set of molecular tools to develop a vaccine. I mean, that's what's to be excited about here, is that these molecular tools are tremendous, look tremendously powerful, and that's something we should be celebrating. Yeah.
All right, not sure if people can hear me, but thank you very much. Awesome discussions, a lot to think about. I noticed that there was a kind of theme going through it about balancing people's opinions currently versus the future promise. But I thought that had an interesting parallel with the what's the right of one generation to impose its will upon the next. And it seems either way, we are imposing our will upon the next, either to have or to not have. So what I wanted to do in this discussion then is ask you guys, in your opinions, how do you balance us as a generation, I myself work in science, especially for scientists, what role do we have in educating the broader public about these technologies in a realistic way, the promises and the risks, to ensure that the laws as they are crafted eventually reflect reality, opposed to, let's say, fears or misunderstandings, either fears, misunderstanding fears or misunderstanding hype, either spectrum. And I'd be really curious to know what you think that is versus, scientists do we respect the public opinion, or the scientists do we seek to change it through education? I'd love to know your opinions.
I guess my view from my experience with public engagement, it's not so much about educating the public, but engaging members of the public in the discussion with the scientists. And one of the things that a project that I've recently finished is this Australian citizens jury on genome editing. And what that involves is actually bringing people together to deliberate over a period of time. So the role of the scientists is to help inform and educate, if you like, people, but to give them the opportunity to get together in small groups and actually deliberate for themselves independently of experts or other people who might influence them. And see, by coming together and talking about their different views themselves, how they resolve these issues. And this idea of citizen deliberation or deliberative democracy, I think, is a really important aspect of public engagement on these issues. And it's really interesting, every single expert report that's been written on genome editing, and believe me, there are many, many expert reports, talks about citizen engagement, but none of them really talk about how this might be done. So we're sort of, those of us who are interested in these things are trying to work out for ourselves how we do this in a way that's properly respectful and so on. And, it amazes me how you can bring together a group of people and they can really come together and understand the technological and ethical and social issues and debate at a really high level. And, they don't have to reach consensus. What they'll do is work out points where they do disagree and points where they do agree. And that to me is really important. And it's really very important that we do it with technologies like this. All of these three people were, as assisting me with this project. I interviewed Rob, Chris and Megan were experts in our citizens jury. And so it's good to bring in experts from all walks of life, scientists, but also ethicists and clinicians and so on.
Yeah, thanks. That's really well said. And thanks for the question. I just want to make one point that, what sort of worries me about today's climate is the increasing sort of polarization and politicalization of issues. And we've seen that, I think, during COVID with science. You see science has kind of become a bit more political, where people are entrenched in their camps, and that's my camp, and I'm arguing from my point of view. And I think that is going to be a barrier to progress and a barrier to actually widespread doing things. I was talking to some people in the foyer before, and I was reflecting of examples of where international collaboration has actually come and made a big difference. And I was reading about, I might get the chemicals wrong, but the sort of CFCs that were burning a hole in the ozone layer. And this is in the 70s, I think, or early 80s, and people came together and they said, this is damaging the ozone layer, we should stop these chemicals, and everyone did it, and we've stopped it. And I feel like now that wouldn't happen. I feel like now there'd be people like, no, we need the CFCs, or, that's, don't believe the data, it's fake news. And, so I think that I want to see science less polarised, less politicised.
I think it's an excellent point, and I think that, again, having forums like this where we can come together and respectfully talk about these things, share views, maybe change opinions, maybe not. But I think have a capacity to have a say. So are there any other questions? Yep, up the back.
Thank you very much. My name is Jennifer Zenker. I'm a research group leader at Australian Regenerative Medicine Institute. And I just wanted to comment it and also end with a kind of question. Due to this very early first experiments on human embryos, that's the reason why we are here today, have such discussion. So we need maybe also to look from that point of view, having seen this kind of feasibility of it, that it's possible, really should also maybe see the positive point on that. And also in order to be able maybe to some extent answer the question of today, where to now, for me, the only way to find an answer is basic research. So if we don't allow any basic research, we will be sitting with exactly the same question here in 10 years, 20 years, and so on. And also regarding the fact that here in Melbourne, we were able to be one of the first people now to create an in vitro system for human embryos, to maybe make use of an in vitro system replicating the human embryo, to take that opportunity and see maybe as a possibility to advance our knowledge. But instead it took over 2 years until we got the rules, guidelines established what to do with these embryos, and they're considered basically as human real embryos now, again limiting us substantially. I'm not sure if that's going the right direction, and maybe you can also have or tell us our opinion on that.
Yeah, I can sympathise with the points that you're making. The challenge, as I've intimated, is that there are a whole array of different views in society about these really important questions. And so I think what the legislation is trying to do is to create an environment where these basic research can be done, but it's in a way that there is quite stringent oversight, which I think satisfies public concerns, or I hope satisfies some public concerns. There are some people who will not be satisfied, as I said, by any research involving embryos. But the choice is not prohibit or be absolutely free to do these things. There's a whole middle ground in there where having appropriate oversight is important, and I think having appropriate oversight is the critical thing, making sure that that works really well.
So again, just to emphasise, I think almost no one says this kind of research should be completely unregulated. But you also, if you think about the scientific method, it's regulatory. Expectations that you make your data available to other scientists, that you're honest, that you don't sexually harass your research assistants, that you don't steal other people's ideas, that's all regulation. So science rests on regulation. Regulation is productive. It facilitates scientific research. Now, of course, the devil's in the detail. And so, yeah, we can have a debate about whether or not it's too restrictive here or too restrictive there. But if someone says, look, I don't think science should be regulated, that's not, they're not being honest with you, because there'll be some experiment that they think, a gain of function, let's see if I can make a super COVID down in my lab for fun, let's I mean, it's kind of weird. People say we don't think this research should be regulated, and then they howl down a scientist who did it on the grounds that it's too early. That kind of public condemnation is itself a form of soft regulation. It's going to be much harder for the next person who wants to genetically modify children now, having seen what happened to the first fellow who did it. So the idea that science, isn't regulated, shouldn't be regulated, that's kind of bogus. What we need to do is have a sensible debate about how it should be regulated, and in that context we should understand all the ways in which good regulation makes science possible, actually speeds research, makes it easier for people to perform certain sorts of experiments because they can rely upon the materials being safe, the results being reliable. So again, devil's in the detail, but if you want to resist this technology, it'll be too late once someone shows, look, this is safe and effective. Gene editing, if someone came to you today and said, look, gene editing's safe and effective and it can allow you to have a genetically related child who won't pass on their genetic condition to their future offspring, that's a much harder thing to resist than the kind of experimental technology fantasy future that we're facing at the moment. So I'm not saying everyone resist this technology. I'm just making the conditional point. If you're someone who worries about a future in which parents are essentially posed with a choice about which genetic modifications they can get for their child in order that their child can get ahead at school, can get the best jobs, has the most up-to-date gene edits, if you want to resist that world, not proving that you can do this stuff safely and effectively would be a good place to start. But again, not everyone, there's costs involved. Let's be upfront. That will also mean that people will not be able to do some of the things that I think people commendably want to do with this technology, allowing people to have genetically related children who won't be subject to various genetic disorders. But again, that's a placeholder for human enhancement when it comes down to it.
Can I just add one tiny addition to your list of things that need to be regulated? To me, one of the critical things as well, of course, is to have science that is trustworthy, that people trust what's going on. And I think we've all focused a lot of attention in our research on what trustworthiness looks like.
Chris, did you want to make some comments before we go to the last question?
Yeah, we're very, very far from the original question, but I think we haven't sort of broached the sort of topic of enhancement and things, and I think it's an important thing to do. There are, as we said, there's diversity of views on this topic, and there are people who think we should use this. We should use this technology any way we can to promote human flourishing, and if that means not, giving ourselves higher IQ, better impulse control, things like that, then we should do it. I'm personally not one of those people. I think we should be using it to stop people dying young. I'm quite a simple ethicist like that. I just think death's bad, especially premature death. Early death's really bad. We should try and stop it. But, I think you can draw, it's a very murky philosophical question exactly where the line is, but I think you can draw some of these lines. And just very quickly, because you asked us to, I'll just address the question of where to now. I think we have to do a lot more basic research now, but just to give, someone who's a bit of a proponent a taste for what I think it could ever be clinically introduced, I think it would someday be clinically introduced in the same way that PGD and IVF is. In the sense that if you've got a family history of a genetic disease that's really, really lethal, you go to an IVF company like you do today, you go and say, okay, we're going to try and create some embryos for you and we'll try and select the healthy ones, great. If you only create one embryo, and if it's got the disease, we'll use gene editing. We could use gene editing through sperm or edit the embryo itself. So I think that is a future, which I don't think will be for a very, very long time, but that is how I think you would see this slowly introduced into clinical practice.
Thanks. Thank you to the panel for such a stimulating discussion. And one of the points that was raised, and it really prompted me to think, was at what point do we stop calling something experimental? And my question is broader than just today's topic. But I think that with any advancement, there is a fine balance between what we already know to de-risk a technology versus what we might not know. And so in a really robust society, how do we balance the fear of the unknown with the hope of the future? Like, how do we sort of meet halfway?
You know, carefully with a lot of community consultation. I do think, I mean, again, as I've emphasised the whole evening, it seems to me people now say this technology is unsafe, and there's good reason to think that's true. But the first time you do it, you're still taking a big leap into the unknown. One reason for thinking that people will be willing to do that is you look at the past history of reproductive technology, where people introduced all sorts of innovations that we still don't have a lifetime of evidence about. So it does seem to me that people are going to be willing to take risks. Lots of people, the decision to have a child is very personally risky. People die, women die in pregnancy. People experience lots of stresses associated with being a parent. Children, terrible things happen to our kids that you can't do much about. So we are always going to be taking risks. In part, this is about some big picture philosophical questions about what things you think human beings should control and what would a world look like in which we controlled them. And sometimes I think the answer is, yeah, that'd be great if we, people don't die of cancer so much or heart disease. You think, whoa, that'd be fantastic. But parents having to kind of make all these decisions about the future of their children by selecting from a menu of gene edits, that's not a world that's attractive to me. But that's a bigger conversation than we can possibly have here.
So Di, a quick comment, and then Luan, one last question at the back.
I just have to get back to basics and get back to the law. So there is a vast body of law on what is experimental and what is standard medical practice, because of course this is relevant where you're looking at negligence. When is a doctor negligent? It's very different if they're using experimental treatment versus standard medical practice. Our colleague Cameron Stewart at the University of Sydney has written vast amounts on this, on how the courts make that distinction. Very different sort of discussion from the philosophical one, but I just wanted to point out that there are legal avenues for looking at these issues as well.
My name is Bob Phelps. I'm with Gene Ethics, and I'm the author of the leaflet that you have all received. And I'd like to say that I think it's been disingenuous of the panel tonight not to have talked about mitochondrial donation, which of course was approved by the Parliament last March. And Monash University has just been awarded a grant of $15 million to begin the research. Now, it's like what Rob said, it's research aimed purely at enabling a woman with mitochondrial disease to have biologically related children. She has other options, and this is clinical research which could lead to live births. Diane is the chair of the regulatory committee at NHMRC, which is going to oversee the research. So I don't take your reassurances about the robustness of the law seriously at all, because we have heritable human germline manipulation about to begin at Monash IVF. The minister announced it last week. So is it justified? The Parliament agreed with the people on the panel among those who convinced them to go ahead recklessly and foolishly. We are already in the midst of this early stage research. What do you say about it? How do you defend your promotion of it? For instance, Rob's project on how do we sell mitochondrial donation to the Australian public, for which your group got $500 $500,000 to do the research on selling mitochondrial research to the Australian community. We need some answers to the things that have been disingenuously avoided tonight.
So look, I do think that you're right. We should have been talking about mitochondrial donation. I think that point is well made. There is some debate about how heritable it is and the nature of the genetic modification, but I think that's a fair point. It should have come up in discussion. I guess because we started with the montage from the summit on genome editing, certainly my mind was fixed on the kind of classic genome editing. I mean, full disclosure, I'm on, I think we can say that, the embargo has been lifted on that. I mean, I'm on the grant at Monash University that is investigating the ethical rollout of that technology, if that's possible. So, so I am implicated there, and I think, again, that point's well made. I guess I certainly don't think that the other grant that you mentioned was that's a fair description of the grant, selling this to the public. I'm not interested in selling any of these technologies to the public. I'm interested in genuine ethical reflection on these technologies, and sometimes one needs to be close to the science in order to do that. But that is something that I think, people should be thinking about whether the same arguments we've been discussing tonight, particularly the obsession with genetic parenthood, which I think is a feature of the therapeutic case for this kind of genome editing, is also that criticism can be made of mitochondrial donation as well. But I think that's part of, I think that's part of the debate, is we do need to be thinking about that technology as well. And to the extent that we've ignored that, I think that is actually unfortunate. Yeah.
Can I just put it on the record that I didn't talk about this because of my position, and I'm just not at liberty to talk about this because of that position. So that's certainly my reason for not bringing it up.
Just a few words, I guess, about mitochondrial donation. So I think it's good to point out that one of the big drivers about the change to the law was actually driven by families who suffer from mitochondrial disease, which can be a devastating disease. It can lead to death in childhood. And people wanted this new technology which could give them the opportunity to break the cycle of genetic disease in their families. And I think it's completely appropriate that you can conduct research, which is all the $15 million is doing. It's conducting research to see if this is a safe technology, which I think is entirely appropriate.
I always wonder whether you'd like to comment also about the experimental nature and how it's presented, and the counselling associated with that's envisaged within the grant. No. Yeah, maybe I'll comment on that.
Megan, I'm happy to talk.
That would be great, Rob.
So I do think this is a good example of something that once upon a time people said, no, that would be grotesquely unethical, it's too dangerous, we don't have enough data. And now we're going to do it and see, it seems to me. That's slightly...
simplistic.
Yeah, no, that is... I was just self-correcting there. That's slightly too flippant. But it is striking how at one stage everyone said this is very experimental. It would be unethical to perform in human beings. We need more fundamental science. And now in the UK and in Australia, that's being rolled back, and people are now interested in using it for the reasons that Chris described. And I think that's one good reason to think that a similar thing is likely to happen with this kind of genome.
Thanks. Can I say one more thing? So thank you very much for raising it. Thank you so much for asking such fantastic questions. I just wanted to make a comment that when we structured tonight, we didn't explicitly ignore Maeve's Law. We didn't explicitly ignore it. It just, there is a lot to cover here, and I want to make a distinction, and I think this is reflected by the scientific community, that there is a distinction between what we're talking about here in terms of changes to the nuclear genome and changes to the mitochondrial genome. We don't have a chance to talk about that today, unfortunately, but I see them very distinct, and I suspect the panel may as well. But I just wanted to make that comment that we did not deliberately avoid that issue, but we have been engaged extensively over the last 5 years associated with that legal reform. And I do think it sets a possible agenda for how we could tackle other reform in the future.
Thank you.